Very excited to be here today. Thanks a lot for having me. And my talk complements the first talk of this session in a way that Yulin was talking about mice, and I will be talking about dogs. But more seriously, I think it answers the question, can we apply these phylogenetic methods to real tumors? Yes, we can, with some caveats. So let's jump right in. So I will be talking about structural variants to make sure that everyone is on the same page. Those are genomic alterations, more than 50 base pairs. And you can see some examples you may be familiar with, such as indels, duplications, inversions. In cancer, we often see more complex stuff going on, such as chromoplex, when there's a balanced exchange of DNA material between multiple chromosomes, or chromothripsis, when the entire chromosome is shattered and then reassembled in a different order. And as you may notice, these events may affect multiple genes of whole chromosomes, which makes it more tricky to draw the effects. By the recent estimate of PICO consortium, about 50% of drivers in pan-cancer are explained by structural variants. In osteosarcoma, the subject of our talk, this estimate is quite a bit higher. At NCI, we have a comparative osteosarcoma program that is led by Emily Blank. And what this program basically recruits dogs all over the country who happen to have osteosarcoma. Why are we doing this? Well, osteosarcoma is a relatively rare condition and in the US, and mostly affects kids or young adults. With 70% long-term survival, there are actually few prognostic biomarkers that are available, and treatment and outcome have not been changed for over 30 years now. In dogs, it is a much more common condition, unfortunately with the lower survival rates. The clinical workflows are pretty different though. In human, it starts with biopsy for the diagnosis, then chemotherapy, then surgery. All the large samples that are available would be chemotherapy treated. In dog, it starts with amputation followed by chemotherapy, so large volumes of untreated tumor are available, and we can use this as a resource to see how the tumor naturally evolving and look into heterogeneity as well. So naturally, we combined forces with the Oxford Nanopore applications team to perform sequencing of 2 dogs. So for each dog, Oliver and Piper, we sequenced multiple tumor sites of the same large tumor, and we have adjacent normal samples. Each sample was sequenced by a single flow cell, R10, shared DNA. We got plenty of data. We were very happy with this data. So here you can see just the coverage plot for each sample. As you can see, there's a lot of copy number changes in heterogeneity going on. So here's well, first we started to look at structural variants, and so those are somatic structural variants in Oliver, and I will explain exactly how we call these variants in a little bit. But what you can see here is there are definitely 2 chromosomes that stand out, chromosome 5 and chromosome 20, and this is color-coded by the number of samples that support the structural variant. So this basically shows you the heterogeneity. On chromosome 5, most structural variants are clonal. We observe them in every sample versus in chromosome 20, most of them are subclonal. And another way to look at this is through this upset plot. And again, I don't expect you to remember the upset plot that Yulin showed you, but I can tell you that they look pretty different. In a way how the structural variants are distributed. So what we see here is most of the structural variants are actually subclonal. So they shared by a subset of samples, but not all the samples. And this kind of naturally, this is consistent with the idea that this is more of a natural evolution of this tumor without bottlenecks that the treatment may introduce. Of course, we can look at methylation, and we can do it in a haplotype-specific way, which is nice. For example, if you look at one particular region in multiple tumor samples, you can see it may behave pretty differently. For example, this region in sample 2 has a hypomethylation in both haplotypes. In sample 5 has also hypomethylation, but it's more localized, versus sample 6 within the same region is haplotype-specific. So we are now also working on combining different types of variation, genomic and epigenetic, to ultimately come up with the best model that explains the evolution of this tumor. But we also have a chance to look in fine detail at some of the most complex events that are going on. So this is an example of Oliver's chromosome 5. And you can see the circus plot that shows the pairwise rearrangements that we see, and they're clustered. Those are actually mostly templated insertions and applications. So what we think this might be happening here is at signatures of something that is called chromoanagenesis, which is a whole chromosome cataclysm where a replication fork stalls, and then it starts producing copies from another DNA material that is just floating around and switching tetraplates, and it basically creates a mosaic of a chromosome. And on the same chromosome, in same dog, we found another signature of a different rearrangement, which is called brachio-fusion bridge. And this has been pretty extensively studied with short reads in the context of cancer, but with long reads, we actually can see more finer details about this rearrangements because we really can look at the base level resolution at the breakpoints. So just to illustrate, so this is an IGV plot. It's the colors represent the phases. So you can see there's untouched haplotype, and the other haplotype has a fallback inversion. We can clearly see the breakpoints. We can see the telomeric loss of heterozygosity, which is expected because we know that this event starts from a loss of telomere. And in fact, in a separate study that is shown here, in collaboration with Michael Dean at NCI, we've been looking at brachio-fusion bridge amplification in the cervical cancer context, and we found some interesting rearrangements at the breakpoints that otherwise you cannot really see with short reads. So now, how did we call all these somatic variants? Well, we developed a new tool that is called Severus. The development is led by Aisha, a postdoc in my lab. The tool is now available on GitHub. I welcome you to give it a try. So it is different from the other current structure variant callers in a way that it was specifically designed for complex rearranged cancer genomes. And it also takes advantage of long-read phasing, so it produces haplotype-specific calls, and it can work with matching normal samples or multi-tumor modes, which we did in the case of the dog project. So unfortunately, the pipeline is on the right. Unfortunately, I don't have time to go into details about each step, but I will show you some benchmarking results. So to benchmark, we sequenced a mini panel of commercially available cell lines that have a matching tumor and normal. We sequenced this with like a multi-omics approach. So we had Nanopore, HiFi, Illumina, and since there's no ground truth available, like in the case, for example, genome in the bottle, we took an ensemble approach. So we combined all the calls, and the calls that are supported by multiple orthogonal methods, assumed to be the truth. So against this benchmark, as you can see, Severus consistently had the highest precision and recall across different types of cancer, across different samples, which was, of course, very encouraging. And it highlights that in the concept of cancer, the signatures of the somatic rearrangements may be pretty different from what we typically see in germline studies, and that requires specialized tools. So this analysis was led by Asher, who is a student in my group. So the conclusions, we sequenced 2 canine tumor with multiple sites, total 12 tumor samples in total. And in fact, it seems to be the first multi-site long-read tumor sequencing, which is exciting, of course. We found many somatic structural variants, many complex somatic rearrangements. Most of them were subclonal, few of them were private to particular samples, which tells us something about the evolution. And we are working on harmonizing all different types of data. And we also are developing new tools, in particular Severus, which is now available, and it works pretty well on the cancer, in the cancer context. So with that, I'd like to thank the collaborators in my group, Aisha and Asher, who are postdoc and student in my group, Amy, who is leading the comparative osteosarcoma program at NCI, Paul is leading the genetics program, and of course the Nanopore applications team, in particular Sergey Aganezov, who did a lot of analysis that I showed you today, and most importantly, our dog patients who made this study possible.